Welcome everybody. It's really nice to meet you all. I'm Alex Connor. I'm going to be the chair of this webinar. So I'm just going to start to say that the Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our biochemistry focused webinar series. Topics in the series include different research areas in molecular biosciences, of course, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. You can just go and see the website for more details if you've got any ideas. My name's Alex Connor. I'm chairing today's session. I'm the Director of Postgraduate Taught Education at the University of Birmingham in the College of Medical and Dental Sciences. I also sit on the Biochemical Society's Committee for Education, Training and Public Engagement, which is why I have the fantastic opportunity to introduce to you our invited speaker today, who is Dr. Jo Rushworth from De Montfort University. Jo is the Biochemical Society's 2020 Teaching Excellence Award recipient. So this is awarded annually to an employee in higher education, and the award recognises individual excellence, commitment to continuing professional development, and of course a commitment to student and colleagues alike. Jo is a very deserving recipient of this award. 4 years after commencing her first lectureship, she was one of the youngest ever recipients of a National Teaching Fellowship in 2018. With 100 nominations for the Vice-Chancellor's Distinguished Teaching Award, which she won in 2016, Jo has championed De Montfort University's inclusive teaching and learning strategy, known as Universal Design for Learning. Her approach to teaching provides flexible ways for all students to learn and to demonstrate their knowledge, including songs, video, podcasts, chocolate-based analogies, Jo, for cellular respiration. Jo enjoys encouraging her students to be active learners using team-based learning, flipped classrooms, flipped feedback, voting using Mentimeter, animal noises, summary sheets. The list is long, I can tell you. Jo is particularly interested in developing teaching and learning methods for large and diverse groups of students. And she's a passionate advocate of co-creation, works with her students to create that learning. One example is appointing a student module leader. I'm not giving things away, am I, Jo? And a team of student biochemistry lecturers together. Jo is a reflective lecturer who enjoys trying out new and innovative ideas and loves to inspire other lecturers to do the same. She devised feedback boxes, which are now used across her faculty to help lecturers make the most of everyday feedback from students, something we all benefit from as academics. Jo applied her novel biochemistry teaching methods to co-create a new postgraduate certificate in higher education, which has seen her teaching, assessment and feedback methods be adopted across and even beyond her university. And that brings me to today. Jo will be presenting her award-winning work with a lecture titled COVID-Directed Evolution of Molecular Bioscience Teaching. Let's analyse the mutations together. During this interactive talk, Jo will share some of her own remote teaching experiences around flipped remote learning, remote lab sim, co-creation, inclusion, transitions, game-based learning and lecturer as learner, as well as asking you to share your ideas and good practice. Please try and engage with the talk and share your own experience with Jo, answering your questions and polls if you can. So additional questions will be asked at the end of the webinar. We have 10 minutes for that, but you can send in your questions during the talk. If you have a question, if you type it in the questions box, it's shown in the image on the screen, which means I can stop talking now. We're not here for me. And over to Jo. Thank you.
Thank you so much, Alex, for an introduction that I don't deserve, but is very kind. I'm just going to prepare to share my screen, and then if you would just be able to kindly let me know that you can see it, I'll just go ahead and do that. Okay. Well, thank you so much, Alex, for the very kind introduction, and I'd just like to say a huge warm welcome and good morning, good afternoon or good evening to wherever you are joining us from today. I understand that last week there were people from over 27 different countries registered with us, so I'm so pleased to meet you all. As Alex said, I'm Jo Rushworth, and the talk I've put together today is around us coming together to analyse some of the mutations that have occurred to molecular bioscience education and have occurred in our classrooms over the last 12 months. So my contact details are down here. Please do feel free to get in touch with me. The work I'm presenting, some of it is my own. Sometimes I refer to other people's practice, and I've tried to put in people's names, photos, Twitter details or other contact details, so you can look up some other people and get in touch with them. Please do share your own ideas as we go through the talk. I've tried to include some QR codes, so you'll be able to scan those if you do have a smartphone, or because the webinar is going to be recorded, if there's something you miss, as I'm going to go fairly quickly and mention some different ideas, hopefully you can either grab a screenshot or wait until the video recording comes out, then you'll be able to go back and scan to get the resources. And for those of you that like a competition and a bit of fun, I've hidden some Easter eggs throughout the talk. So if you want to grab a pen and paper, your first clue is here. You have CAC. You will find similar clues on some of the slides. You'll have to look hard, and we'll see if anyone can solve the problem or challenge before we get to the end of the talk. So for live engagement during the session, like Alex said, if you have a question you'd like to ask me at the end, please do use the facility that Alex has mentioned. And the other thing I'm using today for the first time ever, I like to try something new in a daunting situation, is Slido. So you'll be able to do this now if you have a phone, either scan the QR code on screen, or if you simply go to slido.com, put into the hashtag box biochem. I've got my phone by my side, and I'll be able to see the answers to some polls on my phone, and they'll come up on screen. So you'll be able to do some interactive voting. And you could also use the Q&A box to make some comments that you'd like to make to me live during the talk. And if I'm clever enough to look at my phone at the side, I'll see those too. You've also got in this little menu up here a link to the Padlet, which you had before the session, which invites you to share any of your ideas and practice as the session goes through or after the session. And I'll be around at the main conference taking place in a couple of weeks, and I'd love to catch up with you then. So here is a practice slide. You've got on your screen a Slido poll, so you'll be able to participate straight away in real time, and then we can see where everyone is joining us from today. We have Leeds with an exclamation mark. Fantastic. I lived in Leeds for a long time. That's brilliant. So we've got a huge amount of people from lots of different parts of the world, and I hope you all feel really, really welcome. Lots of people from Manchester. Welcome, everybody. So my teaching journey then, and my disclaimer before we start. I graduated in biochemistry from the University of Leeds in 2005. I then trained as a school teacher, and I was lucky enough to have taught chemistry in Ripon Grammar School for 2 years. I then came back to the University of Leeds to do my PhD as a little bit of a mature student, I guess. So I obtained my PhD in 2012. So I'm still an early career member of the Biochemical Society, still within 10 years of doing my PhD. I then did a short postdoc lab manager role, and then was very lucky to have secured my first lecturing post at De Montfort University in 2014. And as Alex said, I was very, very fortunate to have been awarded my National Teaching Fellowship, and I'm currently an Associate Professor of Teaching and Learning. I feel very, very lucky. So in terms of my own teaching and learning journey, I've been a widening participation student myself. I've been an international student myself. I have been and am a disabled learner and a remote learner. And I guess when I started out doing my PGCE in 2006, this is kind of a biochemical model of how I guess I felt. I didn't have much experience. I tried a few things and failed at most of them because I didn't really know what I was doing. And I had lots of development needs. And I guess if I kind of think about the position I'm in now, 15 years on, I guess I've tried and failed at even more things. I think I have even more development needs than ever before. And I've been lucky enough to have accumulated a number of ideas from other people, mainly my students. So if you've come to listen to an excellent teacher, I'm sorry that I don't think I am one, but I am a developing teacher and a very passionate teacher. And I hope we can have a really exciting discussion today. My acknowledgement slide I want to put up front, and there really aren't too many people to acknowledge, but I just want to say that I'm so truly grateful for everybody's support, to everybody who encourages me and inspires me and mentors me, and particularly to thank my students. And I'll try and acknowledge particular individuals as we go through. So this talk certainly isn't the talk that I would have given this time last year if it wasn't for COVID. So in March 2020, we underwent a rapid move to online teaching. So for me, I was teaching my students in a lecture theatre and in labs in March, and then suddenly the Monday after, I was finding myself doing remote teaching and learning. So that all happened, as we know, very, very quickly to many of us. And I think after that term, when the panic had subsided somewhat, everybody was realising in higher education that actually this is opening up, as Tansy Jessup said in this article, fresh conversations about more effective pedagogies. So that's kind of what I'd like to talk about today, really. So for me and a lot of us, I think, in the sector, we saw this as a golden opportunity for team working and learning and kind of getting through this together. So last summer, myself and my colleagues, TJ Moore and Beth Koyski, set up the website lectureremotely.com. And we set up an accompanying webinar series. So we gave webinars which help staff to adapt to remote teaching and learning and assessment. We host a whole load of other people's resources on the Lecture Remotely site. And if you haven't managed to have a look already, you might find some resources from these 3 people and consortia very, very useful. We're privileged to host on Lecture Remotely the Dry Labs Real Science Network, the Remote Forensic CSI Network, and also the resources from Dr. Dave Lewis. So there are around 100 resources in total, including webinars, how-to guides, lots of resources, all tailored around bioscience teaching and learning remotely, particularly around practicals and final year projects. So you might want to take a look at that. So that was one of the main positives that I found. It was the networking and working with colleagues in the sector to kind of improve teaching practice. So I'd like to ask you at this point, what are the positives that you'd like to retain going forward? So I've listed some here. If there's something that doesn't appear here you'd like to mention, pop it in the Q&A and I'll be able to see that live as well on my phone. So you should be able to vote for these options if Slido is working. There we go. So maybe it's taking people just a little while to log into Slido. So you can either scan the QR code or you can go direct at slido.com. So if there's anything else that isn't here, I think there are so many benefits and we'll go through some of them. You can pop them into the Q&A. So that's something for us all to have a think about. Creativity at the moment is coming out on top and trying out new things. And certainly that's one of the big benefits that I found. So then after setting up Lecture Remotely, the first thing I wanted to do was experience being a remote learner for myself. So I am, as Alex said, an advocate of lecturer as learner. And in the past, I've undertaken Chinese lessons face-to-face and piano lessons face-to-face. But last summer, I signed up for remote Mandarin Chinese lessons. Here's me with Teacher Wei in one of our live classes. And here's me not looking too happy a few weeks ago taking my remote HSK2 Chinese exam. I signed up for remote piano lessons, remote recorder lessons, and I also was lucky enough to have done an Open University course online. And all of this wasn't just about me kind of thinking about pedagogy, it was me thinking about how I felt and my experience as a remote learner. And it was very, very obvious to me that I needed to mutate my classroom in lots of different ways, I felt. So I'll just talk through some of them, and I'd be really interested to know how you felt and how you approached this task, I guess, as well. So for those of you who haven't done flipped classroom before, which Alex mentioned earlier, a traditional method of teaching is obviously giving a lecture and then sending students off to do homework activities. In a flipped classroom, this is reversed. So the lecture is pre-recorded, so the students learn the didactic content before they come to class, and then the classroom is used for active learning activities. So I definitely wanted to use a flipped classroom approach for several reasons. The lessons that I enjoyed most when I was a remote learner were those where I could chat to other people and feel part of the community, get to know the other learners and get to know the teacher. Something I was really keen to mock up, as Alex mentioned, is team-based learning. So if you're not familiar with team-based learning, I really recommend this approach. You can look at Dr. Simon Tweddle and Beck McCarter's resources, which I've linked to by the QR code. So I think that should be Dr. Beck McCarter, I apologise. Team-based learning is where students are put into groups, they come to a session, they do an MCQ test by themselves, then they redo the same MCQ test as a team using one of these fantastic scratch cards, and then they do a problem-based learning exercise. So I was keen to try and mock up that team-based learning approach online, I guess. My own visual disability called Ehrlich syndrome made me think about accessibility. I found it pretty much impossible to learn from a screen, and particularly to learn any kind of didactic content, and realised that it was really impossible to try and make notes from something going on live on screen. So how to overcome that? I definitely wanted to keep formative assessment and co-creation at the heart of what I was planning, I guess. So who was I planning this for? So I teach different students, but I'm going to focus today on my first year biochemistry and cell biology students. So they're a large group of around 270 from all different backgrounds. Most of them are from non-A-level backgrounds. They have qualifications like BTECs and Access. They're wonderfully diverse and just a fantastic group of students. Many were living in Leicester this year, many were not, they were elsewhere. And all of them in this module were learning remotely. So I guess what I was aiming to do then was to give them a really rigorous scientific education that was engaging and that was wrapped up in this pedagogy of care and community, so they could really feel like each and every one of them belonged, they could make friends, and aim high. And I just wanted everybody to feel welcome. So that seemed like quite a task. So to design this kind of different curricula and different perspective then over the last summer, I used UDL as an inclusive lens. So if you're not familiar with UDL, it stands for Universal Design for Learning and was devised by the CAST Centre. The QR code will take you there if you're interested to have a look. So UDL is really a pedagogic framework that enables us to embed flexibility and multiple means for students to be able to engage with their learning, to want to learn, and to demonstrate their learning, so that every learner, no matter who they are, their diversity, can engage and master their subject. So at DMU, some of the ideas around UDL look like this. We want to give students flexible learning resources, so things like multimedia, pre-recorded videos, so lots of people are already doing that. Things like PowerPoints instead of PDFs, so learners can modify colours and modify fonts. And we want to use images and language that reflects and embodies the learner's diversity. In terms of flexible ways to learn then, learners should have different ways to engage. A flipped classroom model allows for active learning, so it is considered to be much better pedagogically. Embedding co-creation, making sure every learner has enough support, but also has enough challenge. And promoting learner autonomy also is important. And then assessments, I'll come to this towards the end of the talk, is a big debate at the moment. UDL really calls for authentic, applied, real-world assessments that don't disadvantage anybody. So we've known for a long time the problems with timed unseen exams, and we know that there's a problem with feedback in the sector, and we should move towards student-directed feed forward as opposed to lecturer-directed feedback. So this gave me a lot to build on, I think. So that they were kind of the principles. So what did I actually do? And then I'd love to know what you actually did as well. I'm just going to keep checking the Q&A. So in terms of trying to build an inclusive and welcoming remote classroom, I guess, for these diverse first year students just coming to university, I would usually introduce my module in 25 different languages, all as bad as the other. I'm not doing that today. But I did that as well, but I decided to do some different things this year. So I recorded a short video for my students, introducing myself and explaining some of the things that I struggle with and find tough as a learner. And then I asked my students to make either a reply video or an audio file or a written piece, which a number of them did. And they introduced themselves, so I knew how to pronounce their names, I knew their personal pronouns, and I knew a little bit about them before they started. The other thing I generated was a student avatar class for this year. So there are around 20 or so students here that I just created to try and enable learners to have kind of a sense of identity among this group, I guess. And some of my students actually then made their own avatar in response. So I'm quite attached to these folks. They all have a name, they all have personal pronouns, and they pop up in lectures and assessments, and I'll show you what they do later on. But it was definitely a conversation starter. And the other thing I did, particularly in the first few weeks of term, so I taught in Blackboard Collaborate. And in Blackboard Collaborate, a student will sign into the session under the name that they've registered with on the system. So I sent out guest links, and as you can see what that does, it allows the student to modify their name, but also to include their personal pronouns if they wish to do so. That certainly made a lot of students feel more comfortable and feel more welcome. Another idea that we used for transitions came from my very dear late friend Dr. Jane Sherwood and her daughter Libby Sherwood, which was creating family groups for first years. I was watching the LTHE tweet chat last night, and I think some people had similar ideas on that. That's a brilliant resource if you're not familiar. So I took the 270 students and they were split across 3 programs: biomedical science, medical science and nutrition. And they kind of felt like a big flock of sheep with kind of no identity really, and they would find it really hard to make friends and to find buddies to work with. I thought. So I wanted to give them a little family group. And Jane and Livy's idea had been divide them into families. So I put them into 20 cousin groups, as I called them, based on their personal tutors. So if you were in cousin group 4, you would have around 15 people who are your cousins, and then you'd be split further down into a subgroup of your siblings, of whom there'd be maybe about 5 people. This seemed to work rather well. So when we taught the students in large classes in Blackboard Collaborate, you can create 20 breakout rooms. So that meant that the students could either go into the breakout room that corresponded to their cousin group if they wanted to work with their cousins, or as the weeks went on, if they fancied making some new friends, they could stay in their randomly allocated group. Most of them made little WhatsApp groups for their cousin group or family group, sibling group, sorry. And also they could use the Blackboard tool to start a virtual classroom, to share files and to have a chat at any time. And they also used these groups to do a remote induction treasure hunt, which I won't talk about now, but if anyone else is interested, just do get in touch. So that was kind of setting up the premise for learning, I suppose. So now I guess it's time to think about learning itself and the way that I wanted to teach and the way I thought learners would learn best online. So a question that's going to come up in a minute is the lecture dead, and is the remote lecture dead? So we know that lectures came about when the literacy rate was very, very low, and typically there would be a man in the village who could read and would read things aloud to the rest of the village. So is that appropriate and applicable for higher education these days is the big debate. If you haven't read this piece by 3 brilliant biochemists at Murdoch University in Australia, have a read. They called it "The Lecture is Dead, Long Live the Lecture." So they were thinking about how we can redefine what a lecture is and what it should be doing. So I think this is a key point of debate then if we're talking about mutating the future of molecular bioscience education. Should we go back to teaching students in a lecture theatre just because we can? Once it's safe to do so, should we be doing it, I guess is the question. So I'd love to know your thoughts. Whether you think, yes, we should be giving didactic lectures, yes, we should actually modify the lecture, no, we should do something else. Clearly, there are other options which don't appear on these slides. But it's something to think about, isn't it? And I'd love to know the conversations going on in the different institutions and the different parts of the sector. So it looks as though at the moment, among the people who are voting, the most popular choice would be to modify the lecture maybe, followed by to do something different. But there are still some people saying, yes, we should retain the didactic lecture. That's really interesting, really interesting. So anyway, so the approach I used was pure flipped classroom. So all of my teaching and learning was done remote online. So I recorded my lecture videos as I would normally do. I did tweak them a little bit, so made sure the learning outcomes were a little bit clearer, made sure everything had a green background to make it easier for learners to read on a screen. And then the other thing that I've used for a long time is what I call summary sheets. So I experienced as a remote learner, it's very difficult to try and annotate a PowerPoint when it's going on live. So what a summary sheet is, it's a pedagogic scaffold which invites the learners to actually process their learning down into something that kind of is a summary, but also it's in their own language, as a knowledge check really to see if they've understood the material. So in a recorded lecture, I might do some teaching about primary structure, and as that teaching is going on, instead of a student just passively listening or passively writing down what I'm saying, they're actually splitting screen, they're watching the video on one half of the screen, and they're trying to summarise that material straight onto the summary sheet. And what that means is they'll know if there's something they haven't understood, because they'll be trying to summarise it and they'll go, oh, I'm not sure about that. That'll get a little star, then they'll come to the live class and we'll be able to get to the bottom of that problem. So summary sheets seem a really effective method to help learners to learn and process information online. In terms of then these active classes then that we're freed up for, active classes I wanted to really be around active learning. So this is one definition of active learning that I like. It's where students engage with the learning process through meaningful activities, and they have to really think and apply their knowledge instead of just passively listening. And obviously, probably we're all familiar with Bloom's taxonomy. So clearly, instead of just being in a passive state where we're trying to just understand or memorise information, we can use the classroom time to get our learners to apply their information to new situations, to analyse, to evaluate, to create, and we can really check whether they've understood what it is that we want them to know. So as a school teacher, I would always lesson plan, and I'd love to know how many other people out there have been lesson planning or have ever tried it. And as a lecturer, I'm ashamed to say that I have fallen out of the habit. So last summer, I resolved to lesson plan again to really make sure that my lessons were all about learners and all about doing. So this document is shared on Lecture remotely. You can grab it if you want it, and please do contribute your own ideas so I can update this document in the next couple of weeks. So this lesson plan is really designed for active learning, and it recommends the use of a starter to engage the learners, recap of the learning objectives, and then some kind of knowledge application or task, some kind of reporting back, and the opportunity for you to actually get feedback on your own teaching, and then a method after class to actually keep your classroom open and keep the learners learning and keep the learning journey going. So what you'll find in this document as well, I can't show everything on the screen because the writing is tiny, but there are lots of different ideas for all these different types of activities if you download the document. So I'll show a few here. So typically the online active learning sessions would start with a bit of fun. So whether it was the yes no game, whether it was singing along to one of Mr. W's excellent songs there, or whether it was noughts and crosses, it was always something the students could engage with. So we'll try a game of noughts and crosses now just for a bit of fun. So you're in two teams now. If your birthday is from January to June, you are a nought. You'll need to remember this. And if your birthday is July to December, you are a cross. So as you can see, the game is nearly finished. I've been asking you biochemistry questions for the last 5 minutes, and the winner is about to be determined. You can guess what I'm about to ask you. We have a little amino acid here. The question is just name the amino acid, but you have to put your nought or your cross before your answer or it won't count. And it is fastest finger first. So are you ready? Name that amino acid. Don't forget the nought or the cross. My students became so competitive with this game, so competitive, and people were so keen to put the answer that I was often forgetting the nought or forgetting the cross. Okay, so we have the nought, then we need the answer. So the amino acid is, so if you type a nought, then just followed by the answer, no need to do separate messages. Everyone's too nervous now for fear of getting it wrong. The pressure. Right, I think the cross was first. This is where I've got to be lightning fast. I think the crosses have it. Well done, crosses. Excellent. So that's the way to have some fun and get the learners engaged, I thought. So then it was time for some active learning and some review of the learning outcomes that featured in the pre-video. So as I say, I was teaching in Blackboard Collaborate. And Blackboard Collaborate has buttons the learners can use to agree or to disagree. So instead of simply asking a question and waiting for someone to put their mic on or camera on, I prefer this whole class formative assessment. So I can gauge if most of the learners are getting this top point wrong, I haven't taught it very well, I need to go over it again. Hopefully they're getting these types of questions right and then we can move on, which was usually the case. Then what we would do is very often I would use the student avatars to come up with a fake piece of work with deliberate mistakes in it. So here Zane has done a little summary of glycolysis, Chantel's focused on the investment phase, and so these things would come up on the screen and then I'd be asking the learners questions. So for instance, what do you think of what Zane's written for step 1? Is there anything that's incorrect? Is there anything you change? The learners could use the chat, microphones, annotating on screen. And in terms of differentiation, if there were learners in the classroom who had already done this material and were super confident, I created what I called an extra readers room. So I gave these students, well, they were open to any student, some extra challenges from Stryer and from journal articles, so they could go off into the extra readers room for a portion of the active learning session and go through those questions with those other learners and then come back to the main classroom. So that seemed to work in terms of being able to provide the support that most students needed and then the challenge if any other students wanted to partake in those challenges. There was also some applied breakout group work. Here's just an example of something that was quite fun. So we had some various celebrities who tried to spell their names using amino acids, and then they wanted to find out, could they find them in a real protein? And if they could, whereabouts might they be found? Lewis Capaldi was very upset he couldn't find his name in a protein. So why do you think that was? So this gave the learners something to really discuss and focus on in their breakout groups and was quite fun. The other thing that I really wanted to keep up, as Alex mentioned, usually we'd be using feedback boxes so learners could give us feedback after every class. Online, I think it was probably easier to do that actually than with slips of paper. So I had an open Menti every session. It was actually open before the session. So if the learners had done the pre-work and they had some comments, they could comment then. But after the session, I would always ask, what did you enjoy? What worked well? basically what didn't you enjoy, what didn't work well, how can I improve, and any other comments. So this is the very first lecture I gave in week 1 of term last year, and you can see that some of the things I've done work quite well, so that was good. I thought, okay, I can keep doing those or develop those, but also you can see the learners did have quite a number of suggestions of things that either I didn't do very well or that I could improve on in the future, and I was able to act immediately on those. So I'd really strongly recommend, if you don't already ask for feedback after every class, do it. It's so useful. So all of this was underpinned by co-creation. So I think co-creation is quite nebulous. It's a buzzword at the moment, and people have different definitions and use it to different extents. It's very flexible as a concept. So this definition I quite like. Co-creation is where education is perceived as a shared endeavour, where learning and teaching are done with the students as opposed to them. And I'll mention a report by a body called JISC, a company called JISC, who operate in the UK, who have recommended in their report in November that we involve students much more closely in all aspects from design to delivery. So quite an interesting example of co-creation that we use, as Alex mentioned, is student lecturers. And the student lecturer concept was actually devised by one of my students, Felipe, who's pictured on screen. So he'd studied the biochemistry and cell biology module the year before, and so he was just coming to the end of that year, and he somehow made the suggestion that he could be a student module leader. So I said, yes, this sounds wonderful, let's do it as a bit of a trial. So in the year 2017-18, Felipe was the co-module leader along with myself, and he did lots of different things. Examples are writing the module handbook, co-writing the module enhancement plan, helping with induction. And the other thing he did is he recruited a team of his colleagues in year 2 to be secondary student lecturers. And they were just absolutely fantastic. They did things like some teaching. I remember them doing some drop-in sessions at the start of the year, which were better attended than my sessions. They had to get extra chairs. It was brilliant. And other benefits, I think, about student lecturers, one is they are real models who reflect the diversity of the cohort. If you Google lecturer, lots of white males still come up, whereas the student lecturers really are representative of the global classroom. A lot of the student lecturers are Gen Z, so they're very technologically savvy. So certainly for this year, they've been extremely useful. And they've got first-hand experience of studying the module and doing the assessments themselves. So it just seemed like such a wonderful idea that we would appoint student lecturers across all of the big biomedical science and joint modules for this year. So we appointed student lecturers, I think it was in the September, just before the start of term. There was a team of approximately 10. I've just squeezed on 4 of their photos here. So Mansi, Rachel, Hannah and Simi were just some of the student lecturers this year. If you want to hear more from them, they're speaking at the Advanced HE Teaching and Learning Conference in July. But just a taster of some of the things that they did. They road tested our materials pre-term. So the student lecturers that I was working with in my module sat down with me virtually and looked at the Blackboard shell, my recorded lectures, some of the assessments, lots of different things, and provided loads of feedback even before term began. They co-created assessments, I'll come to that shortly. They did lots of different kinds of teaching, co-teaching, drop-ins, kind of support sessions. They designed revision sessions, some quizzes, they did revision competitions, some of them produced lecture summaries, as you can see here. And they did lots of subject-based and pastoral support of students as well. And this has been so overwhelmingly positive. Do get in touch if you'd like to try student lecturers at your institution. For me, they've just been such amazing critical friends and made such a huge impact on the mutations in my classroom. I think they have been the best thing that I've experienced this year. So a question then, I guess, who should be engaged or who could be engaged in co-creating the future of bioscience education? Who could you sit down with over the summer once we finish marking exams or whenever your particular term ends? To work within a number of different ways. Might not have to be student lecturers, might be some input into a module, input into a course, input into an assessment. What do you think? There might be other things I've missed off. I'm going to keep checking the Q&A as that's your place to try and pop down any other comments. We didn't pay the student lecturers, it was completely voluntary. So it was open to anybody who wanted to volunteer, and they could do as much or as little as they wanted at any point in time. I will try and work through some of the questions at the end, I think. So there may be other people as well that you already have engaged as co-creators, but I think co-creation for me is something that can't be underestimated. So one of the things that the student lecturers did, and this segues seamlessly into the topic of assessment, is they co-created a CAP study virtual lab assessment. So this was my colleague, Dr. Shabana Kasanbhai, who led all of this work. So please do get in touch with her if you'd like some further details, or I can pass on your details to her. So normally, if the students were able to be taught face-to-face, they would be doing a kind of urinalysis style practical, they'd be pipetting, they'd be using spectrophotometers, they'd be doing a BRS assay, they'd be drawing graphs, lines of best fit, working out unknown protein concentrations, basically. So Shabana used, we used in the module, simulated laboratory experiments. So there are lots of companies that offer this kind of thing. You've got Labster, Learning Science, etc. So we used Learning Science at De Montfort University, and they were absolutely excellent. So Shabana had the students do some simulated pipetting, simulated spectrophotometry use, and she also gave the students some other videos to watch. And basically, she designed a CAP study, which is a case study that incorporates lots of different elements of the module. So the students had to work out what had happened to a particular person who was feeling a little unwell, work out the level of protein in their urine, try and come to some conclusions as a one-page journal article style. So the student lecturers got involved in this, and they actually generated these unexemplars, as we call them. So instead of an exemplar, which is a really perfect example of what the students should produce, it's an example filled with deliberate mistakes. And then what that gets the students to do is to pay really close attention to the mark scheme, try and mark this unexemplar, and then pick out the mistakes and the errors that then hopefully they don't make in their own work. Shabana also did some other really neat things which I'd like to try out myself, which are she used the peer feedback function in the virtual learning environment. So she gave students a mock assessment, and then they could submit these to Blackboard. Blackboard jumbles them up, reallocates them to a peer who gives feedback, and then they're redistributed to the original owner. And she also used a dialogue-based marking scheme with audio feedback. So instead of just marking these things anonymously and just giving feedback saying, well, you got this wrong, she asked the learners to write on the mark scheme, which they submitted with the assignment, what their mock grade was, what their target grade was in this assignment, and then what they'd actually like audio feedback on. So that meant that Shabana in her audio feedback was really able to tailor that to the particular learner in question and to their learning needs. So that went down really well. Other mutations then to assessments, I guess. So as soon as we went into lockdown 2020, I made a curriculum modification to my module. I got rid of the timed unseen exam, which I've been using for years, and replaced it with an online multiple choice question test where the MCQs would be much more applied. So you can see here, these are some mock MCQs. So what I did is every lecture, as part of the active learning experience, students would be given these type of MCQs to look at, and you can see they're not the kind of thing you can Google because they're made up scenarios. Some of them were more fanciful than others. I like doing things for particular occasions, so this was something that was taught around Halloween, I think. If you want to have a look at two really good resources around making multiple choice questions, use those higher order thinking skills and making sure nobody can Google the answers to your MCQs, have a look at David Smith and Beth Rogoyski's resources below. So essentially I kind of wanted to set up this mutant form of team-based learning. I didn't quite crack it, I don't think, but I had a go. So any suggestions gratefully received. So what I did is every lecture there was a very, very short MCQ test, only comprising of maybe between 3 and 6 questions. So the students would have a go by themselves. So you can see I'm so nice. I gave them, in the first week of term, in their very first lecture at De Montfort University, I gave them an MCQ test. Obviously, most of them failed. I hadn't prepared them really about that test, but I wanted to set a very, very high standard going forward in their module. So the students were then able to go away. And we reviewed these things in class as well. They could go away and repeat that MCQ test with their friends or by themselves. Most of them, I think, probably had to go with their friends and talk to their buddies. And then you can see in the blue, when they did the repeat link again, most of them got 100%. And they could repeat the MCQ again and again and again. And the idea was they do it until they get 100%. So what I could see and what the learners could see is they were learning, they were making progress. And there's a lot of dialogue in the HE sector about learner analytics, learning analytics. I think something like this is really neat because what we could see is the first MCQ test most people failed. Within 4 or 5 weeks of term, the class was nearly on a 2-2 average. And these things were very, very tough. The other thing, and this was all shared with the students, the other thing that was shared with the students is the more of these little practice MCQs they did, the higher their score in a much bigger, meatier mock exam that I gave them. So it's really encouraging the learners to engage, and it's watching them engage and kind of reassuring yourself that if students are doing what you're asking them to do, it seems to work. So Professors Sally Brown and Kay Samble have done a lot of work about rethinking assessments during the COVID pandemic time. And they said recently, unseen time-constrained exams have been known to have their limitations for some time. And they pointed out this is a wonderful opportunity for us to rethink assessments across the sector. And we're here today to think about molecular bioscience assessments. And they say we must never revert to the bad old ways again. I've got to admit, when I did my remote Chinese exam, it was really tough. It was very applied. I had to respond to sentences and words and dialogues and things on the spot. It wasn't a memory test. Had it been a memory test, a written exam, I would have been able to prepare all that stuff and just regurgitate it, and I would have found it easier. But I found it fair, and it reassured me that what I'm doing with my students, I believe, is rigorous and I believe is fair. So I'd really like to know your thoughts. Many of you may already have changed your assessments on your programs or your modules or in your teaching. Many of you might be thinking, right, over this summer, I want to change some things going forward. What are kind of your priorities? Do you want to keep exams? Are you going to keep exams? Do you want to make your exam questions more applied? Do you think you could maybe use an open book exam instead of a timed exam? Or do you want to move away from exams entirely and bring in more problem-based kind of assessments or more practical assessments? I'd be really interested to know. Oh, someone's working on the chart. Someone solved half the puzzle. You've solved half the puzzle. If you're the person in the chat, you've solved half the puzzle. Someone's asking about decolonising the curriculum. If you get in touch with me, I can pass on the details. We've actually got Pro Vice Chancellor. If you look up Kaushika Patel, she's led a fantastic project that is all about decolonising the curriculum at DMU. You'll find details of that on the internet, but I can give you those details if you like. So this is interesting. So lots of us actually want to move away from a kind of closed book exam. So I'd love to know what people's plans are for over the summer. And the last thing I'd just like to mention in probably the few minutes that I have remaining before the kind of main Q&A is gamifying the remote classroom, partly because I'm a huge fan of games, board games and puzzles, but also I think that science is all about problem solving, and actually gamifying the classroom is a brilliant way to develop all of those scientific thinking skills and problem-based skills that we want to see in our students. So gamification is the process of adding game-like elements into a normal teaching and learning scenario. So games typically involve things like elements of competition, rewards and points for doing something. So Professor Ian Turner does this really well. That's his field of expertise. Have a look at some of his resources, including this one, Who Wants to Be a Biologist? I will be stealing that next year. Game-based learning is the process of actually creating an entire learning experience that is a game. You're basically building a whole game. So here's an example from a previous dissertation student of mine, Sam, who created, and he did all this work himself, he created a PlayStation game that tested students on their knowledge of organelles. So it's reasonably hard to see in the photo, but you've got the Little Big Planet game up on the PlayStation, you've got different organelles, and there's a character somewhere that you would have to use to grab different organelles based on dialogue that comes up. So there's kind of two examples of what other people have done. So two things that I did then, a bit of gamification. I wanted to motivate the learners to go through their practice multiple choice questions until they got 100%. So this is a bit tenuous. I've called this the CRISPR method of MCQs because I edited my original method, I know. So here's a normal MCQ test, which I'm now calling clues. So I gave this to my final year medical science students, and I could have simply then asked them, okay, question 3, what's the correct answer? But probably then if they didn't get the answer right, the students may not have then gone back and revised the material. So to motivate them, I included a response instruction step. So they inputted their answers into Blackboard, and if they got the question right, they were given a piece of the puzzle. So here they've got a number 9. And if they didn't get it right, they get a little prompt saying, okay, maybe think about this, go back and have another go. And then they have the solve step where they have to try and unlock the padlock. So if they got all 5 questions right and they got all 5 numbers, they'll basically be able to unlock the padlock, which in this case is a password protected Word document. And the reward, I don't think it was a reward, was me running a 5K on a cross trainer because the students had been doing something similar and I was trying to do it with them. So I don't know why they wanted to see me in cycle shorts, but they got to see me and somehow it motivated them. So I really like this idea of CRISPR, of providing a reward or motivation for students to re-evaluate their old MCQ tests. And then finally then, this is an example of game-based learning, and this was all devised by my final year project student Freya. She actually built a remote escape the lab activity. So if you haven't done a remote, if you haven't done an escape room before, have a look at that, and they're becoming much more common in pedagogy and use in the science classroom. So Freya created this, the biochemistry laboratory. Those of you with eagle eyes will already be spotting some clues. So you had to use the clues in the lab. For example, oh, what's behind the Biochemist magazine? There's a crossword. So the crossword was based on the Learning Science lab simulation around spectrophotometry. So the students may have to go back and revise this. And they were doing this activity in teams in small breakout groups. So it was an opportunity to make friends as well. So solve the crossword and you'll know who won the biochemistry crossword competition. That will unlock another box with another puzzle piece. So you've got all these puzzles going on and all these clues, which ultimately the students put all of these puzzle pieces together and they'll complete an Excel spreadsheet, which gives them the absorbance data to calculate protein urine concentration of a different patient. So all of this is preparing them for Shabana's practical assessment. And without spoiling anything for Freya, we are preparing her work for publication. She found that this activity did boost students' confidence, it did boost students' grades in their practical assessment if they did it, and it did so in an inclusive way. And I'll talk more about that another time. So finally then, what was the phenotype of all these mutations? Did any of it work, I guess, is what you might want to know. So Shabana and I had been teaching up until week 5, and we decided to do a national student survey type survey. So we're asking the students to respond to a series of statements measured on a Likert scale from 1 to 5, excuse the disordered numbers, I don't know why that's happened, where 5 is strongly agree and 1 is strongly disagree. So generally, they found the way the teaching was done really did allow them to learn effectively. The recorded lectures were useful, the live classes were helpful, and overall they were very satisfied with the quality of the teaching. I think we might have been on 90% satisfaction overall at the end of the survey, and we had a median score of 5 out of 5. We were just absolutely delighted with it, and our students had worked so hard and just engaged with this tremendously. We did have a really high attendance at our online classes, even 9 AM on a Monday, over 200 people coming consistently, very high engagement. And this comment really reassured me. This is a word cloud of all of the students' comments. But the students said they felt comfortable and they also felt challenged. So coming back to that rigorous pedagogy of care, I think we were actually able to set that up. So what is the future of higher education then? So the JISC report, which came out in the UK, and no doubt there are different reports in different countries that will be fascinating to share. This report, called Learning and Teaching Reimagined, had 9 priorities. I'm bullet pointing and condensing them for the purposes of this almost final slide. But JISC recommended that we prioritise a digital blended approach. We think about upscaling our education. We involve students more closely. We think a lot more about inclusivity and accessibility. And we think about upskilling staff. And those people who got up at 7 AM in Idaho in the US might be more familiar with HyFlex because that started in the States. But a HyFlex model is really more about students being able to choose where they learn, when they learn, how long their degree lasts for. So a model like that incorporating practical skills, it'll be interesting to see where that goes. So I'd like to end by thanking you so much for your attention during, I realise this was a very quick whistle-stop tour of lots of different things. And like I say, I'd be really pleased to talk more either at the main conference or if you just get in touch with me. And I want to leave you with this question to ask you what your vision for the future of molecular bioscience education is. Maybe if you had 3 words, what would you say? And this will come up as a word cloud, and I will leave this poll active, so you should still be able to input into this poll while I hand back to Alex for the Q&A. So thank you so much.
Thank you very much, Joe, and thank you for a fantastic talk. We have got time for maybe one question. We're running predictably ahead of schedule, or behind schedule, I think the right word is. So first of all, thanks. We always talk about teaching being a great way to learn and that peer-to-peer learning works. So student lecturer feels to me like a really smart next step towards that. Before we do move on to question, I just want to point out that, as everybody knows, Dr. Rushworth is the recipient of the Biochemical Society's 2020 Teaching Excellence Award. The award recognises individual excellence, commitment to continuing professional development, and a commitment to student and colleagues. So congratulations, Joe, on the award. We wanted to present this to you in person, obviously, but we are going to have to send it to you via post. We're going to take a picture of it, but what we have is this slide for you until you can get the actual physical award by post.
That is just wonderful. I really feel so overwhelmed by all of this, and I promise to have this inspire me to become a better teacher in the future, and I'm just so grateful. Thank you so much.
You're very welcome. Very, very, very deserving. Right then, we have got a question. Probably time for one question, because I've got to wrap it up by 3. I've been on formal orders, but we can send you the unanswered questions, Joe. And also, if people in the audience want to contact Joe on Twitter, or any of us actually, if you want to talk about the Biochemical Society, contact them, or me personally. You're more than welcome to do that. I've got some questions here. I'm going to read 1 or 2 out. The problem is, Joe, normally I triage them with sensible questions, but they're all sensible, so I'm effectively picking them at random. And there are just so many. I literally am picking them at random because I don't know what to do. Right, let's have a look. That was one I really liked. Yes, my biggest challenge, it says here from Samantha, I don't know if I'm allowed to say it's from Samantha, but it is. My biggest challenge is getting students engaged in activities and learning, and this is a distance learning education institution. Any ideas for students working at their own pace from a distance for engagement? Well, that's a hard one, sorry, Joe.
That is it. Yeah, could you pick thanks, Alex, could you pick a different one? No, that's a really, really good question, I think. And it's something that I reflected on, I think, when I was doing the remote learning. What engaged me, what made me want to do my Chinese homework or to sit and play my piano, and were there days when I just felt like, oh, I don't really want to do this. So I think for me, the sense of community was really, really important. The sense for me of continuing learning or keeping learning going, I guess. My late friend, Dr. James Sherwood, was a passionate advocate of something called the Suzuki method, which is a musical method of education. So she taught young children to play the cello. And what kind of is really obvious to music teachers is the vast majority of education goes on between lessons outside of the classroom. So children, learning an instrument would have a music lesson, but then they'd be practicing every day. So how in molecular bioscience education do we get the students to practice every day? So could there be a suite of learning activities and tasks to do that go around lectures? Could there be things like WhatsApp groups, Facebook groups, Blackboard groups? Could it be that we need to nudge our students every day to say, oh, just spend 10 minutes doing this? And I do think tracking progress is really motivational. So if the learners can see over the weeks they're getting better at something, I think they're more likely to engage. But that's a big question. I think some people will be talking about that at the main conference. I can't wait to see and steal other people's ideas.
Thanks, Joe. Okay, I'm going to ask you one more, even though we haven't got time, because it's a really interesting one as well, which is how, and I like the expression, how do we help colleagues to learn the grammar of active learning? This is Jeffrey Vernon. Jeffrey says, yeah, I have very bright colleagues who cannot imagine alternatives to face-to-front teaching and are sceptical about alternatives. They can't use the grammar to redesign their own teaching, and they don't understand why they would want to do this. How do we, I guess, change that conversation? It's a great question.
That is a brilliant question. I'd be really happy to answer to questions going forward. I don't mind. I've got a load of questions on the Slido, which I'll try to answer as well. But for this one, it's something that I've taught on the PG Cert course as well. And I think it's really difficult. And I've got to admit, had I not been trained as a school teacher, I don't think I'd have this toolkit and I wouldn't have this eye for active learning. I perhaps wouldn't even be aware of it. I might just think I'm a lecturer, so I'm going to go and give lectures. So I think we can forgive people for thinking that, because probably that's what I would have thought as well. One thing I suppose I've used is exercises with staff. So you could present staff with a series of learning outcomes and have them try and design an activity to teach students the learning outcomes. So instead of kind of saying, okay, you've got 5 lectures to teach X, in which case people will prepare a PowerPoint and and do some teaching. Could you sit people down with Bloom's? The thing that I've circulated before around stuff, and you could Google this, is Bloom's taxonomy of action verbs for doing, to try and get students and lecturers thinking more about words that ending ing. So instead of sitting and listening, there are other things that we could be doing in the classroom. And I guess building a community of maybe staff who are passionate and have experience of active learning. So could you build some case studies of staff, and maybe using student feedback and student reps, maybe you could get students to come up with a suite of active learning situations that have worked really well for them. Then other colleagues might start to think, oh, that was really effective, people have really enjoyed that, and that's worked well. That's a brilliant question. I think that's a key challenge, isn't it, really, getting you've got early adopters, and probably many of us here today do these things already, and then we need to convince everyone else that they're brilliant, don't we?
We do. It's a PhD in its own right, that question, isn't it? So I'm really going to have to wrap up. I'd like to thank everyone for attending, and Joe specifically, of course. Please continue the conversation online with us. Follow at BioChemSoc and at PP Publishing on Twitter. We always welcome suggestions for future topics and speakers for our biochemistry focus webinar series. So if you've got any idea for a webinar, submit a proposal. You can find out more about the webinars and propose your webinar and view previous recordings on our www.biochemistry.org/webinars. So join us for the next webinar in our dedicated early career research webinar series, which is called Raising Your Professional Profile, and it's on Wednesday the 2nd of June at 2 o'clock BST. At the session on 2nd of June, this will be chaired by Professor Sheila Graham from the University of Glasgow, and she's the chair of the Biochemical Society, so my boss in a way. We will hear from 4 panelists sharing their experiences and providing advice on how to raise your professional profile. And lastly, I want to highlight that it's more important than ever at the moment to stay connected and engaging with all molecular bioscience interested people. It's extraordinary time for all of us, obviously, in these extraordinary times. But it's an exciting time to join the Biochemical Society community of research and specialists, staying connected and taking advantage of our benefits, which includes discounted registration fees for courses at the Society and meetings, and exclusive access to a wide range of grants and bursaries, and personal online access to 2 of our journals, and more. Visit the website to have a look. I've been a member for a long time, and I sit on a committee. Being friends with David Smith, who wrote a couple of your references, has been a big aspect of that for me as well. So please do join, please join in. One last huge thanks to Joe, and congratulations on your award. Many thanks, everybody. You're welcome. So thank you, and goodbye.